Thank you. We'll see where this goes. I'm not an expert on composting, but if anybody knows me, they know that I like to ask a lot of questions, and I'd like to try to get to the bottom of things and find the truth, This composting experiment, I guess you might call it, started on a trip I made. Well, I had been paying attention to folks like Christine Jones and John Kemp and Graham Sate, all these people that are talking about this biological agronomy, and we're trying to understand soil health and figure out how all this goes together. And there's so many different schools of thought out there, but I went to listen to Christine Jones and Jerry Gillespie in Bladen, Nebraska, and they showed a new, pretty easy method of composting. The idea was not to get nutrient value out of it. The idea was to get the biology out of it and to put the signals back into our soils that inspire more good biology. So it kind of gets to this idea of quorum sensing and things, which is a whole nother discussion. But we'll get started here. I'll show you what we did on our farm, I think. Well, I sent some soil tests to Regen Labs, and you can do a total nutrient digestion test. This is really interesting to me. It doesn't say that all these things are available. It says you have them, okay? And that's kind of this whole notion that we just need to get access to these nutrients. We have them in our soils. We just need the biology and the microbes that can accomplish that. And they're, in large part, they're missing because of how we do things. But, that's some pretty big numbers. Our soil test is showing we got 0 phosphorus available. Well, this is saying I got 1,100 pounds per acre. So, it's this biology, if we can get to this is a big deal. We better figure out what we're doing, right? Price of input costs, what they are. Let's try to figure this out. Maybe we can do a little better job. I mentioned the Jerry Gillespie reference. He's got a wonderful website called, that's the website, but it's called Back to Organics or something along those lines, Soil Back to Organics. Take a cell phone picture of that. Go check it out. All the information that I'm talking about is on there, including some videos of how to do this. You've heard of probably the Johnson-Su method, the Johnson-Su bioreactor. Sounds like a fancy term, but it's a real specific recipe for making really high quality fungal-rich microbial inoculant is a better term than compost. Compost, I think, is a term that maybe we are misusing. It's really nutrient cycling, and we're doing the same thing in our fields. We'll talk about that a little bit. And then I took Dr. Elaine Ingham's soil food web class. Real intense. They teach you how to look in a microscope and count these things and see if you have them. Maybe not practical for everyone, but man, we maybe need to start paying attention to this stuff, because if we don't have these things, this nutrient cycling doesn't work. It doesn't happen. This is a video. We'll see. Maybe you got to turn it on for me. This is where we built our compost in 2019, our first attempt. And in the background is where we feed calves in the wintertime. My brother did all this, so I got to give him credit. He played along with me on all of it. And I'm not very good at running the loader. He's really proficient. Piling manure and oats and hay, oats and peas straw is what we did here. And as he's layering and piling and mixing, the more of that you can do, the better. I was spraying an inoculant into the pile. It's specific for lactobacillus, which is from milk. And it's made, they teach you how to make it on that Jerry Gillespie website, easy to do. It starts out with some grain, and you soak it, and you add some milk, and it curdles, and you save that. Now that's a real rich lactobacillus product. You add molasses to preserve it, and some more water to extend it, and that's basically how it starts. And we're spraying that in there, and I guess we went through it. You can stop it or advance it, please. A little better, and packed it down, and, at the same time adding materials, carbon and nitrogen. It's carbon and nitrogen. You try to accomplish about a 30-to-1 ratio, carbon to nitrogen. Go ahead, and then cover the pile. And you want to get it pretty solid along the base. Old silage pit, right? We used to make silage. I don't know, some people probably still make silage, but we quit doing it years ago. But it's kind of really reminded me of that. And in about October, pulled the back and reheaped it and covered it again for the winter. And I have a sample of that in the back of, it's called static pile compost. And I sent it in for testing, and I have the analysis of what the microbes are in there. Now, they would say if it has the adequate, I think I have a picture of that coming up, but what would be good compost is defined by Elaine Ingram. She's defined it as biologically complete compost. And what that means is you have adequate amounts of bacteria, adequate fungi, adequate arthropods, adequate nematodes, adequate protozoa. All of them are in there. And you want them at ratios based on what you're trying to accomplish. So the further you are along a successional path, the more fungal it would be. And for most row crop production of what we do, we're trying to grow corn and soybeans and oats and wheat and whatever we're trying to grow, it's generally about a 1-to-1 ratio is what we're trying to get your compost to. This couple of pictures of, okay, you have your compost now, how do you get that on the field? Well, you don't have to spread it on there. You can spread it, but you don't have to spread it. Use what's on the farm, old chemical tote, and cut the lid out of it and dumped a load of compost in there, and we filled it with water, and like Anthony showed earlier, this looks like pretty good stuff. This is a video as well. I think we might have to just wait for it to start. Really scientific collection here, I'll tell you. An old bathrobe, and threw it over a tote. This is, it had, I don't think it had sat overnight. It had soaked for a couple hours, however, and, filtered it well, and you can go ahead and stop that. We get the idea. And then the next one, I took it, one more. Real scientific and, high tech here, but pour it into, this is a video as well, just try to get it into a jug and then into the sprayer. And I mixed it at about 25 gallons to 500 water. And I added some other things in there that I'm making some fish hydrolysis and things like that. Sprayed it on the cover crops. We did spray it on some soybeans. We sprayed it on some corn. I don't think we killed anything that we know. There again, I can't say that we hit it out of the park either. But we're just trying to get biology back into these places. And I think maybe the best place to do it is going to be on cover crops. And that's a great place to experiment. That's where Anthony sprayed his. There's our sprayer. I mean, we don't have 120-foot equipment. We got a 45-foot old pull-type sprayer, and it does the job. We might have to think about, if this becomes our agronomy, we might have to upgrade a little. Here's the definition of biocomplete compost, or as Elaine Ingram defines it. And she's been teaching this for a long time. I mean, we thought she was a little bit of a kook, but I heard John Kemp say the other day that a kook is just a keeper of odd knowledge, which I kind of, maybe I fit that definition too. We sprayed it on some beans. I really, like this picture. It was our soybeans last year in the drought. I thought they were, man, these look fantastic, and good corn residue, and it got awfully dry. In hindsight, after testing my compost, it was highly bacterial, okay? Didn't hurt anything, but we didn't really get that nutrient cycling that we're after, right? So this seed was treated with a biological vermicompost treatment, and then I sprayed it once with a foliar. Looked just fabulous, but as the year went on and it got hot and it got dry, I think the benefits of that initial kind of wore off. I don't know this, I'm just thinking about it and what I've learned about this nutrient cycling and when these things come available. So we're not getting a whole lot of nutrient, not a lot going on there, right? It just kind of laid there, all the residue. And everybody talked about this last year, that residue took a long time to become available. Well, the bacteria did their job, but nothing was really happening once it dried out for them to be eaten and provide that nitrogen kick and all those other nutrients when the plant really needed it. So I didn't have the complete biology. That's what I'm thinking is going on here. They yielded, fine as the year was. We needed, another good inch of rain in there somewhere, but it just never happened. And this is Jason Strand's corn taken last fall. In this case, he is adding the carbon and nitrogen all in one place, right? You got it all going on there. The same thing that I put in my compost pile, he's going to have in this field. And I think that might be really what we're trying to accomplish with the cover crops, is get the carbon and nitrogen ratio going, let the microbes do what they do. They assemble and make that aggregate that we're after. And when they're all there and they're all able to do their job as they were created to do, it happens. So I think that this is a backyard composting I'm doing. This doesn't take a lot of acres. I can probably make enough compost in a thing like this to do a couple hundred acres if you're making a tea out of it or an extract. I shouldn't use those terms interchangeably, and people that know this would say, they're two different things, and they are. An extract is really where you put something in a bag and you massage it and get the microbes out of there into that solution. And otherwise, it's more of a I forget the term. If you took the worm compost and just ran water through it, you would have pretty much pure humic acid, which is good product, but you aren't getting the full benefit of the biology, because to get the biology out of there, you got to work that around a little bit. Not so much that you kill them, but you got to get them loosened from the whatever they're stuck to, and they're in that compost. So it takes a little bit of thinking of how you do that best. I haven't figured it out on how to do that in a tote. I'm not going to crawl in there and do it. I mean, we just got to think about, maybe I don't have to make so much at a time. That could be, make 5 gallons at a time or something. That's manageable. As a seed treatment, it doesn't take much. I think one of the recommendations I read was about 1 liter for 1,000 pounds of seed. You don't need a lot of it. This is what we're after here is to get that biology happening right at the root zone, the rhizophagy cycle. You're hearing that term. That's biology is all happening right there at the root, at the grand exchange, as others have said. And this is where I've been using it in our garden. Our garden is in an old sandbox. The kids are all older now, so we don't need a sandbox. So I just started throwing some compost on it, and we didn't bust the bin on garden vegetables and feed the whole neighborhood either. But to see this was exciting to me. It's kind of cool to see this stuff happen. But I want to leave a lot of time for questions too, and I'm sure there's people that are way more experienced with this than I am that could share knowledge about it. I ask a lot of questions, I guess, and for me, it's taken me in a lot of different areas, and I kind of get a kick out of it. But that's probably why I can't hold a job down either very long, so I keep asking more questions. Anybody, if you got a question, shoot it out there. I'd be happy to try anyway. I come from a line of farmers and pastors, so that's kind of a interesting combination. But you talk about having a good attitude, and we heard that the other day or yesterday, and having to how it helps you just take a positive attitude and approach to things. And I just wanted to say something about that. I thought that was a great comment. And, we're all put on this earth for a purpose. And I think farming is probably right at the top of the list. Helping your neighbor, feeding the people. Don't ever be ashamed of that. That I just wanted to mention that. I wanted to say it. So.
Jim, if you go back to that last slide, Jim, can you explain what's going on with the soil on those roots?
No. That's being called the rhizophagy cycle. That's the sunlight-driven process of photosynthesis, bringing that free stuff into the plant, pumping it out of the roots, root exudates, exchanging the carbon and the nitrogen and whatever else is in that free, whatever's coming through there, driven by light, and exchanging it with the microbes for what the plant wants. So, that's going to get your plant off to that healthy relationship, symbiotic relationship, where carbon is exchanged for what it needs, what the plant needs. If the plant has all that given to it for free, none of that happens. So, let the microbes do their job is kind of the idea. We haven't figured this out. I think there's just a huge opportunity for agronomy here to answer and apply this at scale. If Christine Jones and what she's talking about is correct, we better get a handle on how that works. That's how I feel about it. And so we've talked about kind of developing a learning group of people that, maybe you take 10 acres someplace or 5 and just start trying this and see if we can see and learn, more importantly, learn and share. What are we seeing here? And, as we answer more and more questions, maybe we'll have more and more confidence of, spreading that idea that maybe we don't have to be dumping all this stuff on the crop every year. And backing off, it could be a transition tool, really.
Jim, we have a question over here. I was curious if you know of any benefits or I guess the changes in adding dead cattle or other livestock into that compost mix. I know I've heard Gabe Brown talk about the economic value of the different micronutrients and macronutrients that are in a cow, and it's
Yeah.
more profitable to keep those nutrients on the place rather than take her to town as a pound cow.
Yeah.
And sometimes mine die. I don't know about anybody else, but yeah, just curious how that would affect that compost process or what the outputs.
So this anaerobic static pile composting method that we did, and you can look at it back there, the stuff we did this year actually had a cow in it. And just so happened when I dug a pile of that, I tried to get a sample just before everything froze solid, and I hit something hard, and I thought, it's a chunk of ice. Threw it in the pail, and turns out it was the skull of the animal that was in that pile. So it's in in that sample bucket. But that's all that's left of that animal. It just, and I think after 2 years the skull would probably be gone too. I don't know, but as far as the content of the pile, it compared biologically with the others. It was more bacterial, but it had an adequate amount of fungi and bacteria that met the definition. It was in the kind of the wrong ratios. And so kind of the art here for me anyway, or what I'm going to try to do is to take this compost more fungal. And you do that by adding some more fungal food. And I hadn't figured out how to do that through the winter time just yet. I really don't want to heat the pile up again and start all over. I'm thinking we'll wait till spring, maybe reheat the pile with some, oatmeal or oats ground up, maybe some, higher carbon type things, leaves, maybe even some wood chips and things. Yes.
Jim,
I want to
can
move
you-
it more fungal, yeah. So fungi want a higher carbon diet, a more complex diet. You don't want to put the easy stuff in there because it'll go back. Bacteria like the easy stuff, the sugary stuff, the high nitrogen stuff.
Jim, can you explain over here?
Okay.
Can you explain your pallet process, doing your compost in
Yeah.
your pallet, how much you're turning it and how long it's taking you to turn it?
I piled it, sprayed it with my stuff that you can learn how to do from the website, and covered it, and that's it. And I won't touch it till next spring. Maybe turn it then. Yep.
I got one, Austin. We got one here.
I should explain those samples back there. So, and I wish Jay was here, Jay Fuhrer. He's doing the vermicomposting, and he could, he sent some of that in for DNA analysis. And the vermicompost had 450 different microbes. Half of them, they didn't know what they were. So we just scratching the surface on understanding, what these things are capable of. Well, everything in that's been through that worm is probably a good guy. You can count on that. That was something I learned from Elaine. So what I've done is and what Jay is doing, he's taken his compost that he's made, however you choose to make it. There's different schools. Johnson Sioux. This is actually a bokoshi compost. You've heard of that? It's a Japanese method that's been around for centuries of fermenting. And it's what happened in our silage pile years ago. Same thing. It's the same thing. It's a way to preserve. But it would break down then, and what I did with this was run it through the worm bin. So the worms worked it over even further. That's what those three examples. So I've got the one with the skull in it was this year's static pile compost. The other one with a little bit of oats growing in it, I'm trying to make my own fungal inoculant in that pile. So you got to have a living root for mycorrhizal fungi. So I'm trying to get that going there, and then I'll have my own inoculant, hopefully, that I can get across our land. And then, but that compost was also the base feed for the worms. So The worms have, that's 2-year-old compost. It's been through a composting process and then further composted by the worms. And I'll use that as a seed treatment. Probably on all of our acres this year. I don't know, see how far my brother will let me go.
Can I make a comment, or?
Yeah,
Okay, I'm been composting since 2014.
great.
I am doing windrow composting, and I do up to probably 1,200 ton per year. I have also done the Johnson Sioux, and most of the windrow composting is all highly bacterial. So to get the fungal, you need to put it into the Johnson Sioux mix and add the worms to it, and you'll get your fungal material.
Sounds great. I think that's the plan. I mean, and before we try to use it more as a crop input.
But I then take and extract it and put it down the furrow with my seed.
Right. Right in the furrow. You should be giving this talk, sounds like. Because I'm new. I'm really a rookie here.
Jim, when they talk about putting it down with the seed, though, you don't want any fungicide on your seed, correct? I mean, that's got to be naked seed or comment.
I don't have any science on it. You hear various things. Fungicide's a fungicide. I guess I would say that.
I mean, I do it both ways. Sometimes you can't get the untreated seed, and if I just continue to put the inoculant in the furrow with the seed, regardless of seed treatment.
I think if you have a really good biologically complex product, it'll sort itself out. The good guys will win.
So what you're doing, though, is you've got a stream going. You're putting it in furrow in a stream.
I am putting it in furrow,
Yeah.
about 2 to 3 pounds of compost in about 20 gallons of water.
So you can do in the furrow, which we're going to try. You can treat your seed. And those are all good things to use, extracts or vermicompost. Or you can do a foliar application later on, and you can do all three. The foliar is generally an attempt to get a colonization on the leaf of good plant protecting microbes. And that's generally more of a brewing process with air. I haven't done that, but I have sprayed fish hydrolysate and some other products that I've made myself on as a foliar.
I got one. Here we are using an actively aerated compost tea as a seed treatment. So you can take a small amount of compost and stretch it out over a large amount of acres.
That's the idea, yep.
Yep. And we're running about 30 gallons on a 700 bushel truck. So we're soaking that stuff down pretty good. Like, you can't overdo it. Like, we'll have tea running out the tailgate sometimes. So you don't need to put that much on, but you can't overdo it.
As long as it doesn't cause problems,
Plugging
in your
and
grill or something like that.
And
Depends
we
on
have-
your equipment.
Yeah, we have not seen problems plugging or anything with the
Good.
seed fairly wet. Yep.
So there's some experience out there, it sounds like, and this isn't completely new for us, but I think we have so much more to learn. Getting these microbes to work for us, it's exciting to me, what the potential is. And think of the agronomy that could be built around this. I mean, we should have somebody making this stuff within every 10 miles so that the farmers have access to it. Wouldn't that be wonderful?
Yeah, that's very important because like shelf life is 4 hours.
On a
Like
tea,
an
yeah.
actively aerated tea, so it's gonna be important to have that stuff locally made.
Yeah, I don't
close
feel
to the
like we
farm.
can ship this stuff all over the
No.
place. Now, some of the inputs into the, you make a base serum, and then you can put that into another further fermentation. That's actually what I'm using to inoculate the pile. It's about another 3 to 6 week process, and that has some things like seawater that you can make, salt water, kelp. It has blood meal. I mean, there's some other things that diversify the microbial community. That's the whole point. And then you're extending that into this this composting process in an anaerobic. So the pile, if it's working right, it'll heat up and then it'll cool down. And so you're getting two different processes going on in there. One's aerobic and one's anaerobic. Yep.
And when we put together our compost, usually overnight, it'll turn to 140 degrees.
Yeah.
And the main thing with windrow composting is to control your temperature and your moisture. And those are
Right.
the two main things.
Yeah.
So for organic production, it has to be over 130 degrees for 15 days, and then it's considered as organic compost.
Right, and it kills all the bad guys, the pathogens.
And it's supposed to kill all of the weed seeds
And
and
the weeds.
everything into your pile. So
Now, I want to say one of the best comments I had was from Steve Kenyon earlier. I asked him, now, do you use any of these things in your system? You know, he's got incredible results. And he said, no, you know, the best compost tea comes right out of the back of a cow. And I'll agree with that. We aren't shortcutting anything as far as soil health and creating a good habitat. And Dwayne says this all the time. You know, you can release all the pheasants you want to, but without habitat, they aren't going to make it. And we know that. And this is the same thing. You got to have good habitat to get this to work. I think, you know, maybe part of the reason that we're struggling to see, you know, just a wildly successful outcome is we have pretty good biology after, you know, 25 years of no-till. We've got stuff there. And I don't know that we can improve out on a whole lot, but to get the full complement of those critters to where they're cycling that every time a nematode eats a bacteria, it poops out plant available nitrogen, okay? So that's the way it works.
Jim, could you talk about, over here, sorry.
Having trouble.
Could you or the gentleman, whoever, talk about your use of synthetics since you've started this and your profitability level, and if you've done any side-by-side trials?
We would love to have kind of a work group of people to do that. I don't have good numbers. You know, everything we did was just kind of, let's see what happens. And probably did more than I should have. But understanding carbon to nitrogen really didn't sink into my head until I did it. And you realize that, and Dwayne said, he says this too, it's just like feeding your cattle. You can't feed them all carbon, you can't feed them all nitrogen, they don't do well. You gotta feed, and what is a fermentation? It's just like a cow's rumen, it's the same thing. So maybe the opportunity here is for people that don't have livestock, you're getting some of that biology extended from livestock. I mean, there's plenty of manure in this, and getting that out, you know, to get that biology back to somewhat what it should be.
Jim, excuse me, I'll be right there, but Jim, just real quick, when you went over the top, wouldn't you lose quite a bit? I mean, because like your residue, when you sprayed that over your top of your soybeans, did I get that right? That was
No, it really didn't accelerate any decomposition, I wouldn't say. What I was spraying on the top of the beans was more of I'm trying to colonize the leaf. You know, I guess maybe some of that did, you know, there was a lot of bacteria in what I made, so it potentially could have accelerated some of the decomposition, but I didn't see that. I didn't measure it. You know.
If I remember right, you said you were planting oats into something, trying to get your mycorrhizal inoculant stuff. I'm kind of curious why you picked oats as opposed to flax or buckwheat, or if, what was the thought behind that?
'Cause I
was
had
trying to
some.
get in your head on that a little bit.
No, I think all those are good mycorrhizal plants. I took some of this compost last year in ice cream buckets, right? The little ice cream buckets. And I tried all kinds of different seeds. Oats, corn, wheat, barley. They all grew like crazy. I mean, just set them in the window in January, and they grow like mad. I mean, everything you need to grow to get something off to a good start is in there. No, it's got nutrient values too, but I'm not counting on nutrient value. I'm counting on biology.
If you're interested in the Johnson Sioux, you can go to the website and they'll show you exactly how to build a container to do your compost in. And what you add to that is your red wiggler worms, and you can get them from Jim's worm farm.
That's where I got mine. I'm trying to keep them alive through the winter.
Yeah. And the other thing is that composting is Mother Nature working at its finest.
Yes.
It's incredible the day-to-day changes that you see in that system.
Yeah, if nothing else, it's fun. It's just fun to watch. I mean, look at that stuff. Go back and look at that vermicompost, which is in the white tote, and tell me you wouldn't want that on all your land. I mean, it's beautiful.
I was just going to ask if you've ever heard of Dr. Wendy Taheri.
Yes.
And I thought it was interesting. We were talking about oats, and they have an affiliation with mycorrhizal fungi. And
Right.
Dr. Taheri has shown that out of any annual crop you can plant, oats is far and above the way to facilitate mycorrhizal fungi in your soil. So there's a reason old timers used it as a nurse crop, right?
Yes.
Like now we know that. So just
Yeah,
wanted to
that's
mention
a
that.
great point, and I think that's maybe where I originally thought oats was, if I got something easy and confined, that would be a pretty good bet. I don't know if it'll work, but it's growing. It wasn't very high germ oats, I'm afraid.
If I could just make a comment on the Johnson Sioux bioreactor. David has done several tests once he's completed the operation using different starter materials. And the interesting thing is that irrespective of the starter material that he starts with, he ends up with the same family of microbes in the same ratio at the end after a year at the completion process. That is a very interesting
It is.
observation.
Yeah,
So
and I would agree with that.
irrespective of what you start with, you're going to end up with if you do it correctly.
The microbes know what they're doing.
Yeah, and I just might say that they're not very keen on putting in live plants into that bioreactor for some other reason.
Yeah, I would agree. I've made compost in different locations here over the last four years or so, and sent it in for analysis, and it all kind of looks pretty similar. I mean, you're trying to get the ratio, and they say the Johnson Sioux has got the really high fungal component right from the get-go. And so mine is, maybe just a little more processing, and it'll get there. But just if anyone is curious about this, contact Anthony or me, and if you got a few acres, it takes a little bit of time and a little bit of equipment, but ideally, I think if we could have 5 acres in your right beside your regular production, I think we can learn some things. And that'll be the point of it. There won't be a lot of scientific analysis done. It's going to be more like, let's look at it, see what we can see. That's kind of how I like to do science anyway, I guess.
Jim, I'm getting here. We got a question over here.
Okay.
This might be more of a comment. This may be more of a comment. So I'm hearing everybody's questions about how do we get it to have the right end result. I would expect that every time you make compost on your farm, using the ingredients that are present in your farm, you're going to wind up with the same end result. Because all you're doing is adding. There's nothing you can do to create an organism that isn't already present.
I'll agree, yeah.
I can't take oats and put it in soil that doesn't have any mycorrhizae and magically wind up with that fungal organism.
No, it's there,
You know,
yeah.
so maybe the way to get the end result is to test material that you're putting in there and saying, okay, where can I find the right mycorrhizal fungi material to put into my compost where it then can populate and
Yeah.
it can increase the population. If you're short on it right now, well, there's nothing you're adding to it that's bringing
No, it's
that
there.
fungal organism.
It's there in adequate numbers, just
But-
not in the right ratios. It was, so,
But the system works a certain way,
Yeah.
right? And if you add enough of it early, you will get there quicker. That's kind of the
Okay.
thought where I'm going, that maybe, you know, the solution to getting the right end result is to more closely evaluate the microbes that we're putting in there. Not the, you know, the large plant material
Okay.
and other things, but to find out what are the microbes we're adding to this pile. How can I get a source that's rich in other microbes?
right. And a lot of the other, you know, just what you said, and what Anthony said earlier about if you want to get a shovel full of good soil, go out into the trees. Well, there's a whole fungal community working really well in that shelter belt, and take a little bit of that leaf litter and throw it in your compost pile. Get it in there. Along those lines, anyway. There's another thing that I don't want to get too far off track, but Christine Jones is talking a lot about not necessarily the microbes, but the chemical signalers that are in this compost product. And she describes it as, that's why you don't have to aerate it and do all these things. You're not trying to propagate a microbial community. You're trying to capture the base amino acids that are chemical signalers, the way microbes communicate with plants. And when you get that in there, there's this rhizophagy cycle and this quorum sensing action that happens, where microbes don't do what they do unless there's enough of them, and they realize, hey, we're here, let's do it. It's the way disease works too. They don't attack until there's enough of them. But this is the way the good guys do what they do. You know, if you got a nitrogen fixer of some sort, they gauge each other's numbers, and they do that with these chemical signalers. It's complex, way over my head, and I probably shouldn't have brought it up, but
No.
it's worth looking at.
Jim, we got one more question, and we got a break, and Austin's got a guy right there.
Yeah, my question is, you create this inoculant, put on the seed, you apply a fungicide, or you apply a seed treat to your seed, you have a stacked hybrid with certain are we sort of banging our head against a rock? And I mean, we're trying to do the right thing, but we're doing, I mean, how does one affect the other one?
I think there's potential for that, yeah, and that's why, you know, we need to do the work. We need to have the 5 acres where we didn't get in the way. Now, either we're going to find out maybe we didn't eat all that stuff, or maybe we're going to find out, man, that stuff's pretty important. This was a failure. You know, that could be what we find out. I don't know.
Well, I've heard a comment. I chopped silage for this one guy, and he had, the father always fed the cattle. And one day the father come out to the son and said, just what in the hell are you doing? What is different? and the son, and I was chopping the silage, and I chopped it every year, and the guy, well, the only difference we had was, so we used conventional corn this year, and we had Roundup corn the prior year. And the comment is the cattle with the conventional corn were a lot more content. They weren't running the bunk. I mean, it was nothing proven on the whole thing, but there's something that was going on there. We just didn't know what it was, you know. And maybe that's just what I'm sort of getting at with this
Yeah.
here, you know.
You know, I think, and this is another thing Christine talks about, is we've done amazing things in plant genetics and systems and approaches to agronomy, and I mean, no doubt we've increased yields, and there's probably a whole lot more people alive on this planet than there would be without all of that. I don't know, but at the same time, you know, what's next if that stuff isn't working or isn't available anymore? And you know, we've probably bred away from a biological system because we never recognized it. So, you know, maybe that's part of it. Yeah, you know, maybe we need to try some old seed and put it in the system and see how it does. Thank you very much, everyone.